So I'm talking about CT research and how you can use it more to integrate it into your research, because a lot of the questions I get from people is, well, it makes really pretty pictures, but how can we use it in research? So we're going to actually talk about that. So I am Michelle Quigley. I'm a research professor at the Center for Quantitative Imaging, which is part of the Energy and Sustainable Laboratories, which is part of IEE. So the Center for Quantitative Imaging, we have services. So we do computed microtomography from nanoscale to micron. We can do anything that is biological or non-biological. Pretty much if it doesn't have a heartbeat, I can probably scan it. We'd also do measurements and image processing services. So if you want to do stuff like look at image thresholding or improving your quality, we can help you with that. And we have experienced researchers, staff, and CT matter experts. Well, quickly, what we'll have for our equipment, we have a Zeiss X-Radia 620, and then we have a GE Vitome XL 300 Nano Micro CT. The Zeiss is really good at small scale sizes, just so that you guys know. We can go down to about 300 nanometers, but the sample size is very limited. A lot of times we'll say if it can fit in a coffee straw, it is really good to scan. But there's some interesting features. We can do something bigger. We've done stuff like cans of soup or something that size. We can use what is known as Scout and Scan, and that allows you to actually register different scans so that they can be connected. So if you want to look at something at one scale and then go down to another, we can have all those connected. And then we also have a feature called Scout and Zoom, which is if you have a substance and then you want to get an overall view and then something particular that you find, you want to go and zoom in it, we can actually do a quick scan and then actually zoom into that feature that you want. Our GE is our workhorse. It's very flexible. We can go from anything down to even like a half a micron size to 100 micron is our as big as our detector will let us go. But we can fit anything from the size of a bear skull into something small like an embryonic mouse. So we have a lot of flexibility with that. So now let's get into actual stuff. So one of the things CT is great is for pictures and videos. Stuff that will enhance things. For example, if you want to add something really visual to a proposal or a paper, they really work well for that. We can do both videos and images. You can do annotations on these images. So this is a meringue cookie. It's very light. And we have a scale bar in it, but you can also do measurements. So if you wanted to know what that particular pore size is, you could do that. If you wanted to add an annotation, so maybe this thing is a sugar particle. That's my thesis anyways. You can point it out and you can add pictures so you can put nice Penn State stuff on it. The other thing you can do is you can actually do some qualitative analysis things. So for example, the sugar cookie, those big density particles, you can immediately notice that they're very small in the center and they're very big at the outer edge. And that's a qualitative thing that you could look and say, okay, what's going on? Why is this happening? Another thing is this is actually a ratchet wrench and it wasn't ratcheting. And so we actually looked at it and it's not touching where the ratchet is. So it's just spinning. So it can give you some qualitative stuff. There's other things I've done. We've done soil sampling and we scanned it. And then there is one sample that 50 to 75% was a single stone out of the sample, which isn't very useful. And so we knew right away that we couldn't use that for stuff. So if you need some real quick quantitative things, something's going odd, you don't really know what it's doing, a scan may actually be really helpful for you. So what the other thing we can do is we can separate components. And if we can separate components, we can do research on it. So in this case, we have the high density, but we also separated those pores. But you can get really complex. This is actually from Joan Rixmeyer's lab, and they're actually separating individual components of an embryonic mouse. So you can get anything you can separate. We can do measurements on it. And so there's a lot of data that is involved in these images. And you can actually do quantitative analysis on this. So this is actually from those pores and those high density things. So the porosity, about 75% of that cookie was air, which makes sense since it's just whipped egg whites, versus only 1.9 or about 2% of it is sugar, or was that high density, which I suppose is sugar. You can see that there's a lot more, the average volume size, the pores are much bigger than the small particles that were high density. More surface area for actually the small particles, which makes sense since small particles tend to have higher surface areas. You can see a surface area to volume. This min-orth max ferrite diameter and sphericity, those are all measures of how spherical a particle is. And you can see that the pores are more spherical than those tiny particles. Yes, there's a difference. The two of them are measuring two different things. They're measuring it in two different ways. So, but we can also do actual quantitative analysis. So this is actually distribution of the pores and then the volume of those density particles. And you can see that they have very different thing. Even the scales are different on counts. How many sizes? One is 5, I think that's 50 million versus 5 million on the other one. So you can get data from that. We can look at the surface area, same thing, differences. And even that surface area to volume ratio, and you can immediately see that there's way more variability in the pores than there is in the tiny little particles. So there's some data you can pull out of this stuff. If we look at that min-max or the min-ortho max ferrite distance, which is a measure of that sphericity, and the actual sphericity measurement, you can see that even though we're measuring the same thing, we get different results. And one of the reasons is the pore sphericity, small particles tend to appear spherical even if they're not. So if you have a lot of small particles, then you end up with a lot more spherical things. So different measurements can show you different things about stuff too. And this is really visible on the stuff with the really small things, because you can see with that min-max ortho, we have a fairly good amount that seems spherical, but when you actually do the sphericity, because it ends up being a huge amount with the sphericity. I'm sorry, there's almost none with the min-max ortho, which is probably more close to the truth, but because those are very small particles, the sphericity overthinks that they're spherical. So those are kind of the things you can pull out of samples. But what else can we do? Well, CT is non-destructive, and this means we can get 3D structure without cutting. A lot of people in plant sciences are used to doing histology, but you can actually change the 3D structure, and I've actually worked with some people with plants where they've noticed things that they've never seen in the histology because it always gets crushed when the cutting. So that's one way you can use a 3D CT. You can actually use those samples for further analysis. So, and one of the results of this is that you can actually get a record of a sample before you do destructive testing. A lot of people in anthropology, you might have to take, I know I work with somebody who was doing teeth. The whole tooth had to be taken for destructive sampling. So they actually were able to scan that tooth, and then you have data that you can use later. So for example, you want to take measurements of it, you can do that digitally. And so they have a record of that sample, even though it's been destroyed. The same thing, I'm a soil scientist. We take soil samples. We then have to destroy them to take carbon, nitrogen measurements. Not this sample, but other samples I've done. We've done 3 or 4 or 5 different work with those just off of one CT scan. So you can do a lot of things, and it's a really great record to do CT scan stuff. The other thing about CT scans is that you have location knowledge with a CT scan. So CT scans provide a physical location within the sample. And this is really nice because it can actually help guide sampling or other analysis. For example, we actually did this with a graphite sample where they did a large scan, we did a smaller scan, and then they were able to go really small with a TEM and get really detailed samples on a specific section to look at the microstructure and then connect it back to the macrostructure. We also had a client who had a electrical component that wasn't working. So we took a CT scan and they saw this weird blue stuff and they didn't know what it was. And then they were able to pinpoint where that location was, do an elemental map, and actually look what was going on. Other things that I've done is we had somebody who was doing ancient DNA on sunflowers, looking at domestication of sunflowers in the United States. And one of the things, and unfortunately we didn't do the CT scans until afterwards, we were able to see where the seeds were in those sunflowers. And that would have been a really great way for them to get better DNA because the DNA was preserved much better in those actual seeds. And they could have done less destructive testing at a specific site without doing as much harm to these irreplaceable samples. So sometimes doing something beforehand that will guide where your sampling is can really help. So the other thing is you can actually match some of the CT analysis that I showed to actual other analysis. I did this a lot with my PhD work. We'll use our lovely meringue cookie. If you decided to sample over on the outside where those particles are, and then somewhere in the middle where they're the smaller and less of them, and let's say you run CN and you have a higher CN ratio out here and a lower CN ratio inside. Well, we can look at that and say, look, we have these huge sugar particles. There's no nitrogen in them. That's probably what's leading to that higher CN at the inside versus inside. There's less of that sugar. There's more of the meringue, the protein, which has nitrogen. So that would give us lower CN rich. So you can use these to not only find out why stuff is happening the way it is, but you can also connect it to pore sizes and stuff. We've done that. So it's a really helpful thing to do if you want to direct some sampling on your samples. You can actually check location of instantaneous devices. So again, I'm going back to my soil because that's where I'm is. If you, for example, took this core and you said, I want to put something right there because that has all the features that I want to look at. You can put the probe in there and then we can scan it and we can say, yes, you are in fact in that location. So you can go ahead and do your experiment. Or we can say, no, you're off by a little bit, and then you can move it and actually have your probe in this correct place. So it's really helpful for doing that to correct things before you end up doing your experiment and everything goes wrong. Another thing is you can actually compare differences between an object and a model. A lot of our additive manufacturing people do this. This is, for example, this is from Guha's group. And they were having problems with this build where they were getting a lot of powder buildup. And so they knew what it was supposed to look like, so they then can look where it wasn't working and then adjust their manufacturing process to do that. But you can also do that with other things, like if you wanted to do, again, I'm going back to soil, you wanted to look at flow processes, well, you can get your measurements, you can look at what the structure is looking at, like, and does that all fit what your model is doing for what you're doing. So they can be really helpful for that. The last thing that you can do is you can track temporal changes. This is an example that we've done with an additive manufacturer. So they have this design that wicks water up. And so we took a picture, and then over time, we took other pictures, and we were actually able to build a model and a video and watch, you can see the water flow up the thing. We've also done stuff where we've monitored growth of plants. One thing is 2D, we can do 2D really easily. 3D, don't do anything fast. It takes a while to actually get a 3D image. But if you're doing something slow, like plant growth, then we can do stuff. Or if you want to look at something before you do something and then after you do something, it works really great for that. So this is our website. Please contact us. We are willing to help design and help you with CT stuff. The other place where you can find stuff is we actually have a CT users group. It is open to anybody on campus, at least, anybody who's affiliated with Penn State. And it's a great way to meet and connect with others who use CT. If you want more information on our website, eventually a link will be placed there. But right now there's a link to the original news story, and that link still works. And we ask that you register because then we have your email address. And with that, are there any questions?